We're continuing with chapter 10 in Tortora, and that is the classification of microorganisms. Here's what you'll be learning in this chapter. So we're going to take a look at taxonomy, phylogeny. We're going to take a look at the kingdom classification system versus the domain classification system, especially the contributions of Linnaeus and Carl Bowes, actually, in particular. And then we're going to take a look at the characteristics of the bacteria, so the eubacteria, that is, then the archaea and the eukarya domains. Okay, so let's start with some definitions. Taxonomy is the science of classifying organisms. And it's basically, in the old days, it was the degree of similarity, sort of outward characteristics, maybe metabolism characteristics, or shared characteristics of similarity among organisms that would place them in a certain spot in taxonomy. Nowadays, everything is based on genetics. So now we just have actually a more detailed way of looking at the relatedness of organisms. Okay, systematics or phylogeny is the study of the evolutionary history of organisms by definition. And again, things have changed ever since we've been doing high throughput sequencing, and you basically have all the sequences of a lot of organisms at your fingertips. So the study of phylogenetic relationships. So Linnaeus was the guy that started sort of this universal naming system, and he figured there's only two forms of life. They're either plants or animals, and that was in the old days. And then people figured out that there are other forms of life too, such as bacteria, prokaryotes, and fungi. But fungi, they cannot be in the kingdom of the plants because they really don't belong there. And bacteria, they're prokaryotes. So. And it wasn't until 1937, so it's not even 100 years ago, that the prokaryote was sort of introduced as its own kingdom to distinguish cells without a nucleus. And not until 1968, the kingdom of the prokaryote. And then we had in 1969 the 5 kingdom system, and that is now sort of overhauled with the 3 domain system based on sequencing similarities. Okay, so the domain system is mostly what people go by today because it just really is based on sequence similarities, and it says that the archaea aren't any more related to the eubacteria than they are to eukaryotes. And that is saying a lot because previously these two now domains, they were placed into the same kingdom, which they clearly don't belong into the same kingdom based on what we know now about the sequence comparisons. So again, the bacteria, if they were supposed to be in the same kingdom or remain in the same kingdom, they would share a lot more similarities to each other than either one to the eukaryotes. But the archaea, for example, are not any more similar to the bacteria than they are to the eukaryotes. The 3 domain system was introduced by Carl Bowes in 1978, and it was based on sequences of nucleotides in ribosomal RNA. Let's take a look at it. So here's the 3 domain system, and entirely based on sequence similarities. And it says that the eubacteria share their own domain, and that's what mostly we'll be focusing on as part of this chapter. We're going to take a look at the different classifications of the bacteria mostly. The archaea are in between the eukaryotes and the eubacteria, but they are so different from both the bacteria and the eukarya that they get their own domain. And nobody's paying too much attention to the archaea because they don't make you sick, and so I guess we don't really care too much about them. They will be interesting though, and their metabolism is so different, it's so unique, it's really interesting. But anyway, so in the eukaryotes here, all the eukaryotes are in their own domain now, where they used to be 4 different separate kingdoms. We used to have the animals, fungi, the plantae, the plants, and the protista, sort of the single-celled ones, they're kind of more over here, sort of in this area. Anyway, so there used to be 4 kingdoms, now 1 domain for all the eukaryotes, and then the archaea, their own domain, and the bacteria. So this is sort of the current system based on ribosomal RNA sequencing introduced by Carl Woese. Okay, so here be some sort of highlights of the different domains. So the archaea, they are very unique. They are just either high salt lovers, they love it at high temperatures. When other cells are long dead, archaea are still thriving, so in very strange environments actually. Bacteria, we've been taking a look at them, and so we did sort of the profiles of the bacterial cells in the previous lecture, so hopefully you remember some of this, and there's a nice summary on this slide. And then the eukaryotes, here's an amoeba as a representative one. And so here you have more profiles here of the different types of cells. So we talked about the endosymbiotic theory in the last lecture as well. So endosymbiotic theory says that the mitochondria and the chloroplast, the mitochondria would be something like this, and chloroplast of eukaryotic cells, that they actually are bacteria that took hold inside of the eukaryotic cells for protection, but in return they deliver services. And so the chloroplast, of course, that's the site of photosynthesis, so they're doing the job of photosynthesis for a eukaryotic cell, and the mitochondria, they're doing aerobic respiration for eukaryotic cells. But the reason why we think that this endosymbiotic theory is true is because both chloroplasts and mitochondria have their own DNA, a circular piece of DNA that resembles that of bacteria. And so you can do by sequence comparison, you can really show a lot these days. So here, I feel like this is a very similar picture to what we had in the previous lecture already. So the endosymbiotic theory says there's sort of this ancient cell, the early cell. They were branching off into the 3 domains. So we have the bacteria, then here the archaea that started together with the eukaryotes for a little while and then branched off, and then all the eukaryotes. And so the eukaryotes, of course, the unique thing is that they have a nucleus in which to hold and protect their DNA. So here you have a true nucleus now, and then here comes a bacterium along, and one that's photosynthetic, and another one that can do aerobic metabolism, and sort of they start to go into these eukaryotic cells and start multiplying on their own in there, but they also do a service. They're not just like parasitic and they're living there for free, but they're doing a job. The chloroplast does the job of photosynthesis, and mitochondria, they do aerobic metabolism. Okay, so here there is sort of this eukaryotic host cell, and then there's a bacterium inside of this eukaryotic host. So it's kind of interesting image right there. Okay, let's take a look at the phylogenetic tree. So sort of this relationship of organisms to one another. And the phylogenetic tree sort of groups organisms by common properties, and that's based on fossils in the old days. And still, if you have some very ancient organisms that don't exist anymore, that's all you have. Now, if you have some DNA, then you can establish the sequence, and those would be the genomes, and then you can compare the genome sequences. So based on all of these, the work that phylogenetic scientists have done, you can group these organisms and say that they most likely evolved from a common ancestor, and that the species retain some characteristic of the ancestor. So that means that even our cells, they have common characteristics to even bacterial cells. We have some genes that are highly conserved across pretty much all forms of life. Here's some interesting, it's just a slide that shows bacterial communities. They form these rock-like pillars, supposedly about 3,000 years ago. So it's kind of cool here. This would be some fossil records, more fossilized prokaryotes, and these really ancient ones. That's probably all you're going to have to take a look at fossil records and sort of look at their shapes and maybe you can figure out some other characteristics about them. So here, more fossilized prokaryotes. See these rod-shaped prokaryotes, about 3.5 billion years ago. So that's pretty old. Okay, so let's take a look at some more learning objectives here. We're going to take a look at the scientific names. We're going to list major taxa and differentiate. These are important terms for microbiology students: culture, clone, and strain. So we're going to define that and differentiate that. Also, we're going to take a look at protista and differentiate eukaryotic, prokaryotic, and viral species. So that would be our last thing here. So why are we using scientific names? So basically, if you discover a new species of bacterium, so then, or some new form of life, and you're going to publish that and put your name on it or something, who is it to say that somebody somewhere else on the planet who found the same species and describes it and puts their name on it, that you haven't discovered the same thing? And so then who gets to take credit for it? So and that's exactly what happened. Lots of people were describing the same thing but under different names, and it got pretty confusing. So everybody uses their own language too, and wants to establish their own fame, basically. So it got very confusing because too many same things had different names, and it was very convenient and much necessary to come up with a uniform naming system, and this is where the binomial nomenclature comes in, and that is now used worldwide and consistently, so that everybody knows what are we talking about when we're using a certain name. And this binomial nomenclature is just like most of us have a first name and a last name, and so in the same way you have a genus, and a specific epithet, the species name. So a genus and a species name. And so here are some examples of that. So you can still attach your name to something. That is actually fairly often done, but more often than not, you are describing something about the organism with the name that you're giving it. So here is an example that I like very much: Saccharomyces cerevisiae, for example. So that saccharose means sugar, myces that refers to fungus. So this is a yeast, and it's used for brewing. Cerevisiae, it's beer. So that's a name of an organism. Note that the genus name is capitalized. They are all italicized, these names, and the species name does not get capitalized, so it gets small letters. So that's the common convention, that you describe a species with a genus name that's capitalized, a species name that's not capitalized, but both genus and species name are italicized. So that is the convention. Okay, so let's take a look at the taxonomic hierarchy. And this was all started out with Linnaeus, who started classifying plants and animals based on similarities. Now the eukaryotic species, they are very closely related to one another, as we saw with the domain system, and so based on sequence comparisons, that's now established. But let's take a look at the taxonomic hierarchy. So we're starting out with the broadest grouping, and that would be the domain system right now. So the broadest grouping, we either have a eukaryotic cell, we have a bacterial cell, or we have an archaea cell. So that would be our domain. That's our starting point. So here's all organisms. That's everything. Next, more specific, is the kingdom. And so then, of course, the eukaryotes in particular have then the 4 kingdoms that fall into this category, which is the protista, the plants, the animals, and the fungi. So here we have an example of a fungus. So that would be the kingdom of the fungi. Then we are having the phylum next. That's the next more specific grouping. And here is we're looking at an Ascomycota, and then we're just going further and further down here. The class, order, family, genus, species. So the genus and the species, now we have our Saccharomyces cerevisiae, brewer's yeast, defined through all the levels of this taxonomic hierarchy. So what you should know about it is we're starting out with domain, then kingdom, phylum, class, order, family, genus, species. And that you can do that here for, let's say, E. coli. Let's do it for E. coli too. We have the eubacteria as a domain, and this one would be in the kingdom. Well, there's no separate kingdom here assigned. It's just for the eukaryotes that you have the 4 separate kingdoms. So this is for the bacteria, the eubacteria, the domain, and the kingdom is basically the same. And then we have the proteobacteria, and that would be in the class of the gamma proteobacteria. Then we have the order of the Enterobacteriales, family of the Enterobacteriaceae, and then the genus and the species is Escherichia coli, commonly abbreviated as E. coli, italicized, and the genus name is always capitalized. So I think you get the idea here. Okay. So now let's take a look at the classification of prokaryotes. And I'm going to start a new recording for this new section here of this lecture. So stay tuned.